Good morning. Thank you for coming. This is going to be a fun talk, I hope. I want to point out that I'm the speaker, but there are other people involved, Dr. Oleksiak and our graduate student, Samantha, who are responsible for gathering the lots and analyzing the data. So I just want to point out, this is what the data is about. What we have here is temperature on the x-axis and really the metabolic rate of the mitochondria on the y-axis for 141 individuals. And so what each individual is measured at is 5 different acute temperatures. So they're all raised at 20 degrees for 6 months, and then we expose them 5 temperatures and we measure the metabolic rates. And so mitochondrial metabolism measured in 141 individuals. There are 6 individuals that are highlighted just for the range and the means and medians. And in each temperature, each individual is measured at 5 acute temperatures, all in like a couple hours. So our question is, how do changes in amino acid that form the 13 mitochondrial proteins affect temperature-dependent metabolic rates? That's the question. So just to tell you what mitochondria are, in animal cells, the nuclear genomes, all the genes that are inherited from both parents, both the mom and the dad, right? So that's what you typically think about as chromosomes and the nuclear DNA. Tens of linear chromosomes, billions of base pairs, tens of thousands of proteins. And all these proteins catalyze the work that we do. Me standing here, me breathing, everything we do. That's the nuclear genome. Alternatively, there's a mitochondria. Every cell has a mitochondria, almost every cell has a mitochondria, and it has a second genome. The genome is a cellular organelle, like I said, in almost all cells. It's only inherited from your mother. It's a singular circular genome. It only has 16,000 base pairs versus billions of base pairs in the nuclear genome. And it only codes for 13 proteins. So the 13 mitochondrial proteins are a really important job because it's those 13 proteins that form the oxidative phosphorylation pathway. You go like, what? The oxidative phosphorylation pathway is a pathway that's responsible for reducing oxygen to water and creating almost all the ATP for your cells. It is the reason why we move about. Oxphosphate in the mitochondria produces all the energy in the cells, and the 13 mitochondrial proteins, all 13 mitochondrial proteins are a part of that oxidative phosphorylation pathway. If you look at it, you can see that the mitochondrial proteins, 7 are in the first complex, 2 are in the last complex, and the other ones are in the nuclear genes, and that's how that big complex enzyme pathway works. Here's the important point. A critical metabolic pathway that requires interactions between all these proteins to sustain life are unlikely to have functionally significant DNAs because of all the interactions, because of their importance. They're unlikely to have important DNAs. That is, mutations are most likely deleterious and eliminated. Yet, when we look at oxidative phosphorylation in 141 individuals, we have this huge range of metabolic rates. The question is, does mitochondria affect that? So 141 individuals, metabolic rates for them, an incredible variation in this pathway, up to 8-fold difference among individuals. Now again, these individuals have been raised in a common environment for more than 6 months. It's amazing that there's this much variation among them. And so our question again is, how do changes in the amino acids of those 13 proteins in mitochondria affect metabolic rates? So we sequence the whole genome of 141 individuals. We define the variable mitochondrial amino acids that form the 13 mitochondrial proteins. And there are 24 variable amino acids that occur in each in at least 3 individuals. And they form 48 haplotypes. You go, what? So those are the 24 amino acids, and those are the 48 haplotypes. And what you can see is, well, if you look, an individual has a single mitochondrial genome, right? It's not a diploid. It doesn't have mom and dad. It just has moms. And what you can see is that some haplotypes only have a single amino acid in it, the second row. Some of them have 10 amino acid substitutions. Each of those amino acid substitutions define the haplotype. In other words, which amino acids you have defines your mitochondrial genome. And so we can look at the effect of the amino acids. And so there's 13 to 24 amino acids affect mitochondrial rates. And what you can see here is on the top graph, you have amino acid sets. And then you have the partial effect size at how much of the variance is explained by the amino acids. And the large dots are significant. Red is females, blues are males. And the stars across the tops are whether they're different between males and females. And what you're supposed to see is that 13 to 24 amino acids have a significant effect on oxidative phosphorylation rates. 12 of these 13 amino acids are different between males and females, where females are typically less than males. The other thing is you can look at is just the sum of the amino acid change on the bottom graph, B, and again the mitochondrial rate on the y-axis. And what you can see is the more amino acid change you have, the smaller, the lower the metabolic rates in females, but the greater rate in males. It's the opposite effect in the two sexes. So there's a haplotype effect. In other words, if we sum up all those amino acids between them, and this is just, again, the haplotypes on the bottom. There's 48 of them. They're arranged in order. And on the y-axis, again, is just the metabolic rate from the mitochondria. And the big dots are significant, and the little dots are not significant. And then the stars, again, are different between males and females. But 21 of the 48 mitochondrial haplotypes significantly affect the temperature-dependent metabolic rate. 65% of them are in females. 32% are in males. The effect size, how much the variation that haplotypes explain, is about 8% for females and about half of that for males. Females have a larger effect and more effects than males. So, we have an incredible variation in the crucial metabolic pathway, greater than 8-fold difference. That's actually kind of amazing if you don't know it. That's just like a really important metabolic pathway. It's not what you expect. 13 of the 24 variable amino acids have a larger negative effect in females, and the more there are, the larger the effect. 21 of the 48 haplotypes have larger and more frequent effect in females than in males, which is also very strange. So, what do we have? The mitochondria is only maternally inherited. Females only pass it on. Thus, mutations in males are never passed on. If I have a bad mitochondrial genome, my children never see it. Because they never see it, what we expect is larger effects in males than in females. Because my mitochondria is never inherited by my children, I can have mutations. Sorry, one of my children who don't have my mitochondria. It's so unfair. Anyway, that's not inherited. What do we expect? And this is actually really good theory. What we expect is that males will have more deleterious effects of larger sizes. That's not what we found. We found just the opposite. We find the opposite. Female mitochondrial variants have a larger and more frequent effect on mitochondrial metabolism, which we're still pondering. So I asked the first questions, do amino acid substitutions from the mitochondria affect things? Change the amino acids that form the 13 mitochondrial proteins explain a lot of the variation in temperature-dependent metabolic rates, mitochondrial metabolic rates. And surprisingly, female effects are greater than male effects. So what? The natural variation in mitochondrial genomes contribute to individual metabolism. We are different, partially because we have different mitochondria from our mothers. This is unlike most mitochondrial variants studied in humans. When we look at mitochondrial variants in humans, we find inborn metabolic diseases, and we ask, is it the mitochondria? When you look at people with diseases, what you find is that you find almost all the mitochondrial variants have a deleterious effect. What we find is that we find an 8-fold difference in metabolic rates. They're not deleterious or not, they're all naturally occurring. They're among healthy individuals. Those metabolic variants are due to naturally occurring, potentially healthy mitochondria. These data suggest that we can better understand the human condition by exploring how natural variation relates to health. And that's the bottom line. That's the so what. And we got time, you know what?
Thank you.